One of New York University's medical research facilities has lost thousands of laboratory mice to Hurricane Sandy's storm surge, the research setback that could take years to correct, according to scientists. The NYU Oncology Medical Center confirmed on Wednesday that the Smilo Research Center, one of 3 animal research facilities on campus, was adversely impacted by the severity of the flood surge and the speed with which it came on. Animal resource staff was on site continuously to mitigate the damage from the storm, but due to the speed and force of the surge, animal rescue attempts were "thank you for trying verbose. This recording will go away once the product has been purchased." Unsuccessful, the medical center said in a statement. This facility is a barrier facility that is super clean, which restricts the movement of animals in and out of the facility. NYU said it was deeply saddened by the loss of these animals' lives and the impact this has on the many years of important work conducted by our researchers. The loss of the mice was initially reported by the New York Daily News, citing an unnamed source. The building's power failure also took out freezers and refrigerators, likely destroying other biological research materials, the source reported. Outside, "Thank you for trying verbose. This recording will go away once the product has been purchased." Scientists said the consequences for medical research could be far-reaching. "It's really devastating," said Jacco van Riemen, the medical physicist at the Hubrecht Institute in the Netherlands who has worked with laboratory mice. "The problem may go beyond NYU," Van Riemen told Livians. "Some mice are unique, they're just made for certain research," he said. "So if the researchers didn't send it out to other labs, that line is just lost." On the ground, Hurricane Sandy images making a lab mice can breathe several times a year, and "thank you for trying verbose. This recording will go away once the product has been purchased." They reach maturity quickly. But that doesn't mean that it's easy to keep a colony of lab mice going. Scientists use genetic engineering techniques to create and breed what are called transgenic mice, strains where certain genes are knocked out or otherwise altered so researchers can pinpoint genetic variables in development and disease. Creating these transgenic lines can take years. "It starts with lab work to target a specific gene," said Ashley Seifert, who researches tissue regeneration at the University of Florida in Gainesville. "Next, researchers have to insert the altered genes into mice blastocysts' early embryos and "thank you for trying verbose. This recording will go away once the product has been purchased." Then implant those embryos into mother mice that can gestate the new strain. Then the researchers have to make sure the genetic alterations made it through development and into the sperm and egg of the baby mice so that they can breed and pass on these changes. "Simply knocking out one gene so that it doesn't function in the body takes about a year," Seifert told Livians. Many transgenic mice are more complex than that, however, and additional genetic alterations require crossbreeding or manipulating multiple genes. That can take 2 or 3 years. How to rebuild "Thank you for trying verbose. This recording